Good evening, everyone. Welcome you all to week 5 NPTEL live session, which is based on a topic alternative RNA processing and editing. This is Subhasmita Mahapatra from IIT Indore, PhD scholar. Today we'll be discussing few questions and on this alternative RNA processing and editing. Okay, and please let me know if my screen is not visible to you or if I am not audible to you. Okay, let me know in the chat box. Okay, so here we'll be starting the we'll be discussing few questions on RNA processing. In the last session, if you remember, that we have discussed different types of splicing mechanisms, that is post transcriptional modification including capping, tailing, and your splicing mechanism, different types of introns like group 1 intron, group 2 intron, nuclear specific introns, tRNA introns, and how they become spliced out. Okay, so we have discussed everything in the last class. So today I am just we are just going through the questions, and I'll revise you the few things, and we'll know better things like RNA editing, how is it done, and what are the different types of enzymes are included in it and all. But most of the questions you will see that basically on the B section questions of CSR net exam. If you trying to appear, or if you have appeared CSR net exam, then you may find these questions quite similar to the section B, not C or A. Okay, so please try to answer these questions. It's okay if you don't answer, we'll be discussing things. Okay, so here is your first question of today's session. What is the average size of an exon? 150 nucleotide, 300 nucleotide, 1000 nucleotide, or 100 nucleotide? Which one is will be? Please try to answer this question in the chat box. Then we'll be discussing the answer. As people are joining, I need to admit them. So don't. I'm sorry for the disturbances, but I can help it. Okay, let's discuss it then. As I see, all of you have answered 150 nucleotides. So this is the correct answer. That is 150 nucleotides is the average size of an exon. So can you tell me what is the size of an intron? So here is the thing actually. So introns are of different sizes. We cannot say that intron is of to 200 nucleotides or 300 nucleotides or 10 to 20. It depends upon the organism, and it just changes according to species also. Okay, so exons are the coding sequences, right, and introns are the non-coding sequences. So these non-coding nucleotides have been removed out using your splicing mechanism that everyone knows, right? And introns are, specifically, if someone asks you that what is the size of an intron, then you can answer it as the introns are of variable sizes. They changes according to species. Okay, and the biological advantage of alternative splicing is that two or more polypeptides can be derived from a single gene. Okay, so now you know that human genome, that human, in case of higher organism, if we see that we have a long chain of DNA as the genetic material, and if we just complex organisms require lot of proteins for each and every metabolic processes inside our body. So if we require a lot of proteins, that means we need to carry a lot of genes, right? Because genes leads to production of mRNA, then that leads to production of your protein, and that becomes post translational modification happens, which leads to different types of enzymes and all, right? So in that case, to reduce the this hurdle for a complex organism, this alternative splicing is specifically a boon. Okay, that means a particular, a single chain of a gene which is approximately, for example, 100 nucleotide, it can give rise to multiple proteins, not a single protein but multiple polypeptide chains, which can be further added, like A chain to C chain or A chain to B chain, or like this, in a random manner to produce different types of proteins. As you know that polypeptide sequence is the primary structure of protein. After that it leads to secondary, then tertiary, then quaternary. So if we talk about a quaternary structure of a protein, for example hemoglobin, here you can see this alpha globin and beta globin chains are present. That means two types of polypeptide chains are present. So how come two types? Because this is a quaternary structure, and this, in this case of two types, this alpha chain has been derived from a particular gene and beta chain has been derived from a different gene. So here we can say that because of alternative splicing, a 100 nucleotide sequence of mRNA can give rise to both alpha and beta chain. Here I am taking it as an example. Okay, don't write in your exam like here I am just giving you one example, like one sequence or one single gene, sorry, single chain of nucleotides can produce multiple polypeptides. That's the advantage of having introns. Okay, because generally people think, and we also think, that these are non-coding sequences. Why they are already present inside the coding sequences? They are just hampering the all these processes, because in case of prokaryotes it is very simple, but in case of eukaryote we are getting lot of hampering, and lot of proteins have been used, lot of ATP, your body energy has been used. Okay, so this is the one advantage of your intron, right? Next is, this allows an organism to carry fewer genes in its genome, right? So in case of our we humans carry 23 pairs, right? So 23 pairs of chromosome. So for example, a mice or any simple organism or a very small organism, they they can carry a fewer amount of genes in the genome, and which lead to the different types of polypeptide production. Okay, so this is a chain of your DNA. This is your promoter sequence, and promoter is required for RNA polymerase binding, right, in the transcription process. And this is your gene which has different colored exons, and between them are the introns. And this is the pre-mRNA via transcription process. Next, this final spliced mRNA can contain a multiple sequence of polypeptides. If you see here, that there are four different color chains, that four polypeptide chains have been arranged to produce a single protein. So this protein can be useful in any different type of processes. But because of having introns, here we are getting a single polypeptide which can be lead to different polypeptides. Like if we combine these two, then it will be chain protein A, or if we combine these two, it will be protein B. Okay, so we'll be discussing the splicing mechanism again in the upcoming slides. So try to understand what I am saying, that this different polypeptide chains combine to produce the protein, right? Okay, I guess everyone understood what I am trying to say. So here is your next question. When pre-mRNA is spliced more than one way, it results in truncated RNAs, incomplete RNAs, RNA isoforms, or RNA concatemer. Everyone try to answer this question. We'll be discussing this in 2 minutes. Okay, let's discuss it then. As I see that most of you have answered C, that is RNA isoforms. Okay, so let's see. So this is your answer, that is RNA isoforms. Okay, so let's discuss about all the options. Okay, truncated RNAs. What are these truncated RNAs? So what happens when this pre-mRNA, which we call generally nascent mRNA, right? In case of pre-mRNA, when splicing occurs and uh, at the end we are getting such a chain of mRNA which lead to a half polypeptide chain, or we can say is at as it is, like at the end we are getting a stop codon. Okay, I am just trying to draw here. For example, this is your 5 days to 3 days pre-mRNA. Okay, p-mRNA. It's hard to write. Okay, which will lead to production of your spliced mRNA, or we can call it as mature mRNA. So in case of mature mRNA, what will be there? There will be one capping and tailing process, right? So after this capping and trailing process, what happens? This mature mRNA will be transferred to cytoplasm for translation process. Always remember, thus only the mature mRNA which has been gone through the post transcriptional modification move to the cytoplasm, not the pre-mRNA. Sometimes people get confused between which mRNA is moving to cytoplasm, if the splicing mechanism is happening inside the nucleus or the cytoplasm. So don't get confused between these two things. Okay, so what happens if this is your pre-mRNA, this is your mature mRNA. Okay, I am writing M. After that, when it will lead to production of a protein using your translation process, ribosomes and other enzymes. For example, here we are getting a stop codon like UAA. Okay, so if we are getting a stop codon here, for example, if your protein should be this much length, this is your regular protein, but you are getting a stop codon at this point of your protein, that means your polypeptide chain is not complete. Okay, and also because of this UAA codon, you are getting your polypeptide chain is certain have its length. Fine. So in case of this, we can call it as truncated RNAs. Truncated RNAs means they have been cut in half, like some stop codons are there which is hampering the total translation of a whole polypeptide chain. Got it? In case of incomplete RNA, also it is kind of similar to truncated RNA, where we are getting different stop codons inside this, or you can call it as a so for example, you are getting a sequence of UUU or a GC rich sequence which will lead to production of your uh, hairpin structure of RNA at in the beginning in the middle of translation process, such that the translation process will not get completed. That will lead to incomplete RNAs. Or RNA isoforms, we have already discussed. These RNA isoforms are different lengths of pre-mRNA, which will lead to different types of polypeptide chains, and these different polypeptide chains can combine together to produce different types of proteins. Understood? Now let's talk about alternative splicing, as this question is basically based on alternative splicing. The potential for an increase in phenotypic diversity without increasing the overall number of genes. Fine. If you see, if you talk about mice or Drosophila melanogaster, which is a good model organism for different types of experiments, and also guinea pig. So in this case of model organisms and all, you see that there are different types of genes which, in case of mice, if you take it as an example, that in before going to your human level of drug investigation, we always perform experiments on the mice at first, because we say in the different model organism mice is the best suited for checking out if this particular drug is hampering the human genome process or human body or not. So now you can call it as how much genome similarity is there, but the number of genes and the structure of this mice is very small. Okay, so now how this process been happening, that in case of a small organism we are getting a diversified range of genes and diversified range of proteins. In case of Drosophila melanogaster also, if you see these things, there are very good examples explained in your lecture videos. If you have any questions regarding this, then you can go for those videos. Actually it has been really explained well. Okay, so the potential of an increased phenotypic diversity without increasing the overall number of genes. So number of genes would no need to increase the number of genes to get a highly diversified number of proteins. We can go for alternative splicing, which will help us to understand or increase the phenotypic diversity. Okay, like your different types of phenotypic diversity, you can take it as example your coat color, hair color, or everything. For coat color in case of mice, hair color, or in plants the height of the plant, or drug resistance, or different types of drought resistance or pesticide resistance, or whatever it is. In case of bacteria it may be your, but in case of bacteria it doesn't happen, so we can leave the bacteria. It is achieved by altering the pattern of exons that are spliced together. Okay, we have discussed about it, so I don't want to spend extra time on it. Different proteins can arise from a processed mRNA from a single gene, right? A single gene can produce different types of proteins if we combine them in a specific manner. If someone having any problem regarding this sentences or points or about alternative splicing, then you can let me know. I'll discuss about it again. Okay, next is alternative splicing can occur either at specific developmental stages or in different cell types. Fine, so it is not equal for in every organism. You can call it as a specific developmental stage. Like in there are different stages of in from fetus to embryo to a small baby to then a young boy, or then after the adulthood, or then the old stage. So there are different developmental processes which I told is about human. So it can differ in case of different species or different organism, and also in different cell types. It depends upon which cell is performing which function. Depending upon that, alternative splicing takes place its role. The calcitonin gene yields an mRNA that synthesizes calcitonin, which is a most required hormone in case in your thyroid, or also calcitonin gene related peptide CGRP in brain. So this is a very good example of this. One gene can produce two proteins, like your calcitonin, which is acting, which has important role in case of your thyroid hormone balancing. Also this calcitonin gene related peptide CGRP, which has crucial role in your brain also. So one gene producing two proteins which has different role in different parts of your body. Okay, so this is a very good example of alternative splicing. You can take it or remember it. Two proteins with distinctly different functions, right? These two has two distinctly different functions. One is acting at brain, another one is acting at in your thyroid. Okay, so what is the main function of your thyroid? Anyone? Main function
Growth
of thyroid?
hormone, ma'am.
Yes.
Growth hormone, ma'am.
No, not growth hormone. You can tell, you can say it as it regulates your basal metabolic rate, or you can just call it as a BMI control. Okay, and also it regulates different types of hormones level inside your body. Okay, also helping in growth or growth. It is a part of its role that it helps in growth, but growth hormone is not called the thyroid hormone. Understood? So you can't just tell that it is similar to thyroid, but thyroid has specific role in the growth of a person or a baby, but it is different. Growth hormone is different. Okay. Next is your this alpha
Okay, ma'am.
tropomyosin mRNA have at least 8 different alternatively spliced alpha tropomyosin mRNAs. So what is this alpha tropomyosin? Where is this alpha tropomyosin is found? Which part of your body has this alpha tropomyosin? Yes? Can anyone tell me? This tropomyosin is present in which part of your body? Is it present inside your brain, heart, lungs, kidney, muscle? Yes, very good, muscle. Okay. This alpha tropomyosin is crucial, has crucial role in muscle contraction. It is present inside your muscle. Okay. If you don't understand what to tell, then you just see that myosin, it is always related to muscle. Okay, no need to get so confused between the things or different types of proteins. Okay, so myosin means protein, sorry, muscle. The alpha tropomyosin mRNA have at least 8 different alternatively spliced alpha tropomyosin mRNAs. Okay, so if we talk about a particular single mRNA of alpha tropomyosin, it is generating 8 different types of spliced alpha tropomyosin mRNAs. So these are the very good example of alternative splicing. You can remember it, or you can just go for other examples. Okay, here is your next question. Introns are never seen in eukaryotes, fungi, prokaryotes, or plants. Try to answer this question. We will be discussing the answer in 2 minutes. Okay, so I see most of you have answered C, that is prokaryotes. Okay, this is your answer, that is prokaryotes. Introns are never seen in prokaryotes. Okay, someone said D, but plants do have introns. Okay, remember this, that plants also includes under these complex organisms, and they produce different types of proteins to face challenges in every day, right? So sometimes it may be the pesticide resistance genes, or it may be the your drought resistance gene, or it may be simple function for photosynthesis, for your different functions have been also happening inside the plant. Okay, so you can't say that plants do not have introns. Plants have introns. Okay, so prokaryotes do not have introns, right? Like everyone said this. So now my question is, uh, do prokaryotes have the transcription or translation process occur in a simultaneous manner? Like this transcription is happening, and after that this translation is happening right away, or is there any specific modification been happening after transcription in case of prokaryotes? Do the transcription and translation process occur simultaneously? Yes, very good. Okay, so yes, the prokaryotes, in case of prokaryotes, there is no nucleus, and that's why the transcription and translation process occur simultaneously. Always remember this. In case of eukaryotes only, we are getting such diversified range of post-transcriptional modification, post-translational modification, and all this kind of mechanism, but prokaryotes do not have this type of things. Their translation machinery just started away after completion of, or we can say that transcription is happening when the chain length increases, ribosomes get attached to other part of the chain and start the transcription process, so that the RNA or the genetic information will remain intact. There will be no loss of information, and the RNA will not get destroyed. Fine, so these are few points or few plus points in case of prokaryotes having this transcription and translation in a simultaneous manner. Understood? Fine, so there are four major types of introns generally present and can be distinguished on the basis of their mechanism of splicing or characteristic sequences. We have discussed these things in previous class, but I am repeating it again so that you will get a clear idea of splicing, introns, exons, and all these things. If you have any doubt regarding this, you can ask me. Okay, group 1 introns in fungal mitochondria, plastids, and in rRNA in tetrahymena, that leads to self splicing, where there is no require of adenosine triphosphate, that is your energy molecule ATP. Okay, so in case of self splicing, there is no requirement of any ATP. Group 2 intron in fungal mitochondria and plastid, they also don't require any ATP. The process is called self splicing. But if we have already discussed this, but if you recall it, that group 2 introns produce a lariat structure of intron. Okay, but in case of group 1 intron, simply the introns removes out and exons get bound to each other and form the mature mRNA. But in case of group 2 intron, we are getting a lariat structure of intron. Fine, we will be discussing each and everything in detail. Introns in pre-mRNA, which is spliceosome mediated, they require ATP. Which intron requires spliceosome mediated splicing? We have already discussed in the previous class. Just try to recall it. Otherwise, it's okay. If you can answer, then it will be good. Which type of pre-mRNA or which type of introns require spliceosome? Human? No, no, we cannot call it as a human. Okay, there are different types of introns have been classified, and if you recall it, it is basically called nuclear pre-mRNA. Okay, remember this, we have discussed it in the previous class. Group 1 intron, group 2 intron, nuclear pre-mRNA. Next is your introns in pre-tRNA. Both pre-tRNA and nuclear mRNA, they require ATP. Okay, and in case of nuclear pre-mRNA, we require spliceosome. What is spliceosome? Spliceosome is basically a ribonucleoprotein, that is a combination of RNA and protein, and there are different subunits of protein are present. It is not a single protein, and also you can call it that it is a combination of ribonucleoproteins. That means at first a single RNA and single protein will make a small ribonucleoprotein. Okay, for example, this is one ribonucleoprotein, this is another ribonucleoprotein, this is another. So these three will combine together. Okay, so this can be called a spliceosome. Understood? Any and a good example of RNP or snRNP. snRNP means small nuclear ribonucleoprotein. Okay, and good example of ribonucleoprotein is ribosome. In case of ribozyme, the RNA acts as a catalytic power. RNA has the catalytic power, but in case of this type of spliceosome, it is a combination of snRNPs. snRNPs means small nuclear ribonucleoproteins. There is no catalysis have been happening. Okay, so don't get confused. Fine. So these are the different four types of introns we have discussed so far, and spliceosome mediated splicing also we have discussed. So let's move to next question. Percentage of alternatively spliced genes in humans: 10, 65, 35, 75. Which one is it? If you don't know the real answer, then you can guess it. Okay, let's discuss it then. So I see most of you have answered D. So here is your correct answer, 75%. Okay. I think these questions have been discussed in your previous live session. That's why you all are getting the correct answers only. No problem. We'll be discussing things in a different way. Okay, okay, okay. Fine, fine. Fine, fine. Okay. So here is the next question. Rate of nucleotide synthesis is highest in humans, Drosophila, E. coli, or fungi? Okay, so all of you have answered C, that is E. coli. Okay, this is your answer. Rate of nucleotide synthesis is highest in E. coli. Now let's discuss about nucleotide synthesis. At first, let's start from the basic. So what is a nucleotide? So nucleotide is combination of three things, right? One is your ribose sugar, second thing is your nitrogenous base, third one is your phosphate group, right? So this is a combination of nucleotide. If we combine only the ribose sugar and your ribose sugar and nitrogenous base, then it will be nucleoside. Fine. So this is the basic thing, everyone knows about it. Now let's talk about how these nucleotides get synthesized. There are two types of pathways generally. One is your Salvage pathway, another one is your de novo pathway. So this de novo pathways and Salvage pathways are two precursors of uh, formation of nucleotides, where they are recovered and recycled. In case of de novo, as the name suggests, the nucleotides have been produced from scratch. Scratch means from carbon dioxide, water, and all. Okay, from simple molecules, the nucleotide have been formed. That is your de novo pathway. But in case of Salvage pathway, generally they are recovered and recycled from nucleotides obtained from cellular environment. So if there is one damaged nucleotide or any nucleotides which have been deaminated, or for example in your waste process this have been removed, then it is recovered and recycled through the Salvage pathway, while the de novo pathways, they start the production of nucleotides through your from the scratch, that is simple molecules, carbon dioxide, water, and all. So here is my question. What do you think? Okay, which pathway requires a lot of ATP or lot of energy? De novo pathway or Salvage pathway? What do you think? Which pathway requires a lot of energy? Now we have discussed that there are two types of pathways of nucleotide synthesis, and one is starting from scratch, another one is just recovering and recycling. So yes. Okay, very good. So generally de novo pathway requires a lot of energy because it is starting from simple molecules. Okay, so there is one example here how these nucleotides have been produced, like using glucose 6 phosphate NADPH, which is an high energy molecule, leads to 6 phosphoglucono 1,5 lactone or 6 phosphoglucolactone, you can call it, where water have been added in presence of gluconactonase. This is one enzyme. Always remember when there is one ASEH, that means this is an enzyme which just cuts or the breaks the protein. Okay, now it becomes 6 phosphogluconate. Earlier it was 6 phosphoglucolactone. Next, after that, this is actually a completely long process of de novo and salvage pathway, but here I have just extracted a image where it just what we can call it as, very simply it has been defined. Using NADP, this ribulose 5-phosphate have been produced, which can be divided as xylulose 5-phosphate and ribose 5-phosphate. This ribose 5-phosphate then used in production of your nucleotides. Got it? So we don't need to go through the de novo pathways and salvage pathway in a deep manner, or understand each and every enzyme and mechanism, because we are talking about RNA processing and RNA editing, so we should focus on that only. So here just a question arises, so that I just thought we should discuss about it. Okay, so let's talk about RNA splicing again. Key reaction in RNA splicing is condensation, hydrogen bonding, transesterification, and coupling. Which one is it? Okay, let's discuss about it then. I see that most of you have answered C, that is transesterification. So here is your answer. So let's discuss about each and every option clearly. First one is your condensation reaction. What is condensation reaction? So let me tell then. I am trying to explain. If you don't understand, then you can ask me. So in case of condensation reaction, specifically what happens when where release of water molecules been occur. Okay, like A, this is a product, plus B gives rise to C product plus water molecule. Okay, so this is an example of your condensation reaction. Condensation means when two molecules are combined together to produce a single molecule. Okay, so and also releasing one water molecule. So what is the opposite of condensation reaction? Is hydrolysis, where water molecule is used to hydrolyze. Hydrolyze means one product produces two different products. Like for example, A and B is combined, or if you can just arrow mark it in a reverse manner, that C have been hydrolyzed to produce A and B. In presence of water, hydrolysis reactions occur to cut or damage one product or break one product into two different products. Okay, so now you understood about condensation reaction and hydrolysis reaction. Next one is hydrogen bonding. As the name suggests, there must be one hydrogen bond which connects two different molecules. Okay. Next is your transesterification, which we are going to discuss. Next one is your coupling. Coupling means also here also you can say this is a reaction which is in organic chemistry where two molecules combine to produce a third molecule, or we can say it as AB. Okay, so A is a different product, different molecule. B is another different molecule, but they both of them combine to produce AB. So this is one example of coupling reaction. Fine. So this is the example of transesterification, which is a major reaction been happening inside your splicing mechanism. I am going to discuss about the splicing mechanism again. Try to focus. If you don't have, if you have any questions about it, then you can ask me. Okay. During RNA splicing, the first step, the 2-days hydroxyl of an adenosine residue at the branch point attacks the phosphate bond at the 5-days splice to release 5-days exon and to form an intermediate lariat in which the 5-days end of the intron is attached to the adenosine at the branch point by a 5-days 2-days phosphodiester bond. What is written here have been explained in the figure. If you see the 2-days OH, okay, OH means hydroxyl group. So this 2-days OH group of adenosine at the branch point, here we have been talking about branch point and lariat. That means we are talking about group 2 intron splicing, not group 1. Fine. From the branch point, this 2-days OH group have been attacking at the 5-days exon position or 5-days splice site, you can call it. So from the 5-days splice site to release the 5-days exon, matlab the breakage of phosphodiester bond been happening. Okay. So here the breakage of phosphodiester bond been happening, and I believe that you understand the phosphodiester bond formation. Okay, because this is a part of combining two nucleotides. Okay, if you just try to remember this, then it will be easier to understand how this bond breaks actually. Okay, so this OH group been attacking at the 5-days splice site to cut or just break the phosphodiester bond and release the 5-days exon. That means this exon has been released. As you can see, there is no bond, and for example this GUA point have been removed. That means it is just overhang, and at that point it forms another phosphodiester bond at the 5-days 2-days phosphodiester bond. Here you can see the 5-days 2-days phosphodiester bond. Okay, this is the first step of group 2 intron splicing, where the branch point 2-days OH position been attacking at the 5-days site so that the phosphodiester bond breaks, and the GU or AG, whatever it is, it just forms the phosphodiester bond here at the branch point of adenosine with adenosine. During the second step, the 3-days hydroxyl of the 5-days exon. So this is 5-days exon of 5-days exon attacks phosphate at the 3-days splice site. So here is your 3-days splice site, right? This is your 5-days splice site, and this is your 3-days splice site. So at the point of 3-days splice site, this 5-days 3-days OH group from here, 3-days OH group from here attacks on the 3-days splice site and just break the phosphodiester bond here. After that, there have been a releasing of another lariat shaped intron, and the spliced mRNA have been combined together. Understood? So this process is called ligation. Ligation means another phosphodiester bond is produced which just combines the 5' exon to 3' exon. So if you just overally we can say it as that there is a transfer of your phosphodiester bond from one point to another point, which can be called as transesterification reaction. Okay, understood? Because if the formation of phosphodiester bond is there which combines two nucleotides in the process of splicing mechanism, this phosphodiester bond is been breaking at one point and then been re-established at another point. So basically it can be called as a transfer of your phosphodiester bond, which is at one point can be called transesterification reaction. Understood the splicing mechanism? Is it okay? Clear? Okay then. Let's move to our next question. R loop hybridization paved way for detection of split genes. Is it true or false? Try to answer. Okay. So as most of you have said A, that means it is true. Okay. So what is R loop hybridization? R looping was first described in 1976 to 1977. R looping studies from the laboratories of Richard J. Roberts and Philip A. Sharp showed that protein coding adenovirus genes contain DNA sequences that were not present in the mature mRNA. Okay. Roberts and Sharp were awarded the Nobel Prize in 1993 for independently discovering the introns. Later, introns were found in a number of eukaryotic genes such as eukaryotic oval albumin gene. So, discovery of split genes, how did this happen? In the background study, adenovirus has a DNA genome and makes many mRNAs. So, let me ask you one question. Uh, in a recent this COVID-19 pandemic, adenoviruses have been used as a carrier of for the vaccine production. So in the market, if you see, generally Covaxin and Covishield, these two types of vaccines were very common inside India. So which vaccine contains the adenovirus vector? Covaxin or Covishield? Try to recall and don't Google it. If you remember, then you can answer it or you can guess it, but you know, if you go for Google and then answer it here, then it's no point. Very good. Okay, that is Covishield. Okay, and most of us, if you have heard about it, that people are talking about like Covishield vaccine is more effective than Covaxin, so we will be going for Covishield only, because this is one adenovirus vector. But remember it, this adenovirus have been used for vaccine production. It is very good, it have effective, shown very effective results worldwide. But if in 1 in 1 lakh people or 1 in millions of people, if this adenovirus gene get mutated, okay, in certain time period, like for example 10 years, 20 years, or whatever it's the time period, like if this adenovirus gene get mutated and has this carcinogenic property, then that will be very harmful for that person. Understood? So everything has this plus point and minus point. Okay, so this is just a type of discussion. I am not getting any point about this vaccines or whatever. Okay, so adenovirus has a DNA genome and makes many mRNAs. Fine. Can we determine which part of genome encodes for each mRNA by making a DNA RNA hybrid? Next comes the experiment. They isolated the adenovirus genomic DNA and isolate one adenovirus mRNA, hybridize, and then look by EM at where the RNA hybridizes to the genomic DNA. Okay, this is the uh, figure here, and the outcome they saw that the RNA is generated from 4 different regions of DNA. How can we explain this? This is called RNA splicing. Understood? So if they tried to hybridize a DNA, genomic DNA, and one mRNA, so only from this A, B, C, D portion, at one part of this DNA, this hybridization happens, and they saw that it is been generating from one part of DNA, not the whole part. Okay, so this is basically called RNA splicing or generation of split genes. Understood? That was about R loop hybridization. Any base in an RNA can undergo editing. Is it true or false? Any base in an RNA can undergo editing. Okay, let's discuss it then. At first, we'll be discussing about RNA editing. Till now, we have been talking about this RNA splicing, alternative splicing. Now, let's talk about RNA editing. What is it? A usual form of RNA processing in which the sequence of the primary transcript is altered is called RNA editing. This is basically can be called as changing the RNA sequence after transcription. Changing, okay? So this is different from RNA splicing. In case of always try to differentiate things. Don't get confused between RNA editing and RNA splicing. In case of splicing, we are just splicing out the introns and connecting the exons using your ligation method. But in case of editing, we are just changing the RNA sequence for our own benefit. Understood? So here we are talking about RNA genome editing. Got it? So this happens after transcription. Two types of RNA editing occurs. First one is your base modification, where adenosine or cytosine, these are two bases, two nucleotides, get deaminated. Deaminated means NH2 group have been removed out. Fine. Next one is base, that is uracil. Uracil insertion and deletion. In case of RNA, uracil is replaced, okay, in place of thymine. That is, thymine is replaced by uracil in case of RNA, which is present only in DNA, that is thymine. Okay, so base uracil insertion or deletion, this can be in a both way. Fine. So here I'll be discussing a few part only. You can go for it from the lecture videos as the time suggests. I am trying to cover and in a briefly manner. Okay. So here we will be talking about this insertion and deletion of nucleotides by RNA editing, where we can use a guide RNA in 20S RNP or editosome that is encoded elsewhere in the genome. Part of the guide RNA is complementary to mRNA in vicinity of editing. If you have heard about genome editing using guide RNA or sgRNA, in case of CRISPR-Cas technology, this word have been used multiple times. So I think we'll be also discussing about CRISPR-Cas and all this, but let's leave that thing to future. But so here the part of guide RNA is complementary to the mRNA in vicinity of editing. Vicinity means at the nearer to editing place. For example, this is one example of your insertion mechanism. Here at this GGUC sequence, I am trying to insert certain genes, that is uracil. Okay, only this uracil genes can be inserted and deleted using the guide RNA. There are different ways of insertion and deletion using the vectors, and cloning methods are already present. You can go for that. Okay, but in case of this particular method which we are studying right now, we can insert and delete the uracil nucleotides. Okay, insertion of uracil at this point. Okay, so what is the first step for it? For example, if you want to insert something inside the DNA, then DNA or genome, we need to first break it. Okay, and for the breaking of this phosphodiester bond, we require endonuclease. Okay, next is this endonuclease or exonuclease, which one it will be. If we are talking about a specific sequence at the inside of RNA, then we require endonuclease. But if we require insertion or deletion at certain point or end, like for example, I want to insert a sequence of U at this 3 days end or a 5 days end, then I will require exonuclease enzyme, not endonuclease. But endonuclease are specific to sequence and inside the RNA. Got it? So here there is there is this break where GUC is nowhere present. Now if we see that this CAG have been added already, right? Because you know guide RNA means it is forming a complementary strand to the mRNA vicinity. So this is my guide RNA which has CAG sequence. This has formed hydrogen bonds GC, UA, CG. Now you can see that this sequence has AAC. So to A, which one is complementary? U. For A, there will be the opposite complementary base will be uracil. So now for this A, there is a single U, and for this another A, there is another U, and G and C has been combined. Understood? Now you can see this RNA ligase comes into picture, where it forms the hydrogen bond, also the phosphodiester bond, and complete the strands. So if we look at the initial figure and the final figure, we can see that there has been 2 U have been added. Okay, so insertion of a single nucleotide or multiple nucleotide can change the whole protein synthesis, mRNA transcription, all these things. You know that, right? So next thing is deletion. If we want to delete this three U's from our RNA of interest, that is 5' to 3' nucleotide sequence. Always remember, RNA is single stranded, that's why it is shown as a single black color string, and this blue one is the guide RNA. So here also we are getting a guide RNA which is complementary to the vicinity of your target sequence. So here your target sequence is UGG. For that I will just make one guide RNA like this. G will pair up with C, GC, then U will pair up with A. Okay, so this one is forming 3 hydrogen bonds. Now if you see, there is already single U is present, and there is no nucleotides in between them. So if we see here that this triple U is just added either way. Now I require this endonuclease enzyme which will cut this RNA from at the point of or specific sequence. Now this UDES-exo, there is another enzyme. This UDES-exonuclease enzyme will remove the uracil bases. Okay, now this uracil bases have been removed, and after that you will form a hydrogen bond with A, and if you can see here from the first figure and the last figure, this uracil have been removed. Understood? So deleting or inserting certain few nucleotides or a single nucleotide can change the protein synthesis process, transcription process, and everything. Okay, it can, for example, if you may ask, like, why we are just adding this U and all, my reply would be like, if we just try adding the U, maybe it lead to a stop codon which will just stop the gene and will not produce the protein so that the organism can survive. Or if it is a toxic gene or carcinogenic synthesis, or sometimes it happens, so in that case if we just use the guide RNA to insert or for the RNA editing process, then that will lead to shutdown of that particular gene so that the organism can survive. Okay, this type of examples you can see in case of plant genome editing. Okay, there are very good examples already present in plant genome editing, where you see using agrobacterium and using different other pesticides and all, this have been used, this guide RNA interference, genome editing. Okay, and it has been fruitful also. Fine. So this is all about RNA editing, where insertion and deletion of U, that is uracil, been happening. Next, first one was adenine or cytosine deamination. Second one is base uracil insertion and deletion. First one has been done. Next is base modification. How is it done? Just remove the ammonia. NH3 group have been removed, where cytidine will produce uridine and adenosine will produce ionosine or inosine. See, so these are enzymes, deaminases are required for this, and in this case of base modification also, this is a process including RNA editing, where base have been modified so the nature of the gene can be changed. Okay, so we have been, I believe that RNA editing is clear to everyone. Is it clear? Do you understand the RNA editing process? Insertion, deletion, and base modification? Okay, thank you for answering. Next question is, mRNA alternative splicing always brings truncated protein. Is it true or false? Try to answer this question. Okay, I see some of you have said true, some of you have said false. So actually it is false. It Here it is written always. mRNA alternative splicing always brings truncated protein. It is false. Okay, it doesn't bring truncated protein always. Sometimes it happens. Like for example, in your DDX47, an RNA helicase had multiple alternative splicing events, including one that was more common in B cells, where an intronic region after exon 3 is spliced in, resulting in a stop codon, which I was talking about in the previous slide. Like if there is a stop codon present, and sometimes because of this splicing mechanism, if this stop codon suddenly arises, that will lead to a truncated mRNA, so that it will not get a complete length of your polypeptide chain. That would lead to a truncated protein lacking the ATP dependent RNA helicase, DEAD dead box, and C terminal domain. So this is one example of your truncated protein production. Okay, but not always it happens, sometimes it happens because of stop codon appearance. Got it? Let's move to next question. Prokaryotic RNA polymerase has only one subunit. Is it true or false? Okay, let's talk about it then. As most of you have answered false, so it is your answer that is false. Prokaryotes use the same RNA polymerase to transcribe all their genes. In E. coli, the polymerase is composed of 5 polypeptide subunits, 2 of which are identical. 4 of these subunits denoted as alpha, alpha-dase, beta and beta-dase comprise the polymerase core enzyme. So this is an RNA polymerase example of E. coli, where there is this starting region minus 35 region, and there is this minus 10 region containing tata box TATA, and it is the promoter region. After that your transcription site starts, and in the promoter region at first RNA polymerase binds, and further this transfer of RNA polymerase happens to your transcription site. Okay, and we have discussed all these mechanisms, so you can go for previous session videos which is available in the YouTube to understand it more. Okay, next, select the conserved nucleotide pair at the splicing junction: GCAT, GTAG, ATGG, or TATA. Which one is it? Okay, it is splicing. See, we have already been discussing about RNA editing, alternative splicing. So, and we talked about this adenosine branch point also. So, I am asking, what is the conserved nucleotide pair at the splicing junction? That means where this splicing mechanism happens. And this region is conserved by certain nucleotides. Try to answer this question. If you don't know, then you can just guess it. Okay, as I see, most of you have answered A, but your answer is B, GTAZ. Let's talk about this GTAG rule. What is this GTAG rule? If you see, uh, try to go for the lecture videos. It has been explained clearly in the lecture videos. I am just briefing it about GTAG rule. So if you see here, this is the exon point. This is the 5 days exon site. This is the 3 days exon site, and there have been the introns. Okay, so from which point and what is the conserved nucleotide region? So this has been found that GT, that is guanosine, thymine, and your adenine and guanine. So this GTAG is highly conserved. That means describes the presence of these constant dinucleotides at the first two and last two position of introns of nuclear genes. Okay, So in case of nuclear genes, you will find these two dinucleotides as conserved regions in case of in at the splicing junction point. And splice sites are sequences immediately surrounding the exon intron boundaries. Okay, so this is the splice site from point of this. Okay, so this will be removed. This is the intron, and splicing junctions are recognized only in the correct pairwise combination. So if this combination is not there, like GC is there or something, sometimes it is AT or whatever it is, if it is varied, then it will not be recognized by the spliceosome mechanism. Because in case of nuclear pre-mRNA, spliceosome is required for splicing mechanism. As we have already discussed about spliceosome and the spliceosome mediated reaction mechanism, you can go for the YouTube videos to understand it more, because we have discussed in last class. Okay, so remember this GT and AG, these are two dinucleotides which are present in the nuclear genes, and this is highly conserved. Okay, let's move to next question. Which pathologies result from mutations in proteins of splicing mechanisms? Spinal muscular atrophy, retinitis pigmentosa, myotonic dystrophy, and all. Everyone try to answer this question. Okay, let's discuss about it then. So here is your answer, that is all. Okay. Pathologies resulting from aberrant splicing can be grouped into two major categories, that is mutations affecting proteins that are involved in splicing. That examples are spinal muscular atrophy, retinitis pigmentosa, myotonic dystrophy. There are the three examples where you will find the mutations in proteins involved in splicing. Okay, next mutation is affecting a specific mRNA or messenger RNA and disturbing its normal splicing pattern, and which is very common in case of beta thalassemia, Duchenne muscular dystrophy, cystic fibrosis, Frazier syndrome, and frontotemporal dementia and Parkinsonism. So I believe that these all the diseases are quite relatable, because we have been reading about this from a long time, like beta thalassemia, cystic fibrosis. So now you know what is the reason behind this particular disease. Okay, so there have been mutations affecting this specific messenger RNA also, which disturbs its normal splicing pattern. That's why these diseases occur. Okay, next one is, which RNA specific enzyme is specific towards tRNA? This is the last question of today's session. Try to answer it. Okay, so I see some of you have answered A, some of you have answered D. Let's talk about the enzymes that is responsible for RNA editing. Okay, so these are the enzymes which we've been discussing about. That is ADAR means adenosine deaminase acting on RNA. We have been talking about RNA editing. So there are two process of RNA editing. One is base modification, another one is uracil insertion or deletion. Okay. So in case of adenosine deaminase, what does it mean? That means base modification. Okay. Adenosine or adenosine will be deaminated to produce inosine, and your this cytidine will be deaminated to produce uridine. Okay, next is your ADAT, that is adenosine deaminase acting on tRNA. Next is CDAR, that is cytidine deaminase acting on RNA. So if we are talking about tRNA specific editing enzyme, so this will be ADAT. Okay, understood? Now let's talk about this RNA editing enzymes. So if you see, there are different sequence length have been given here in this figure, like ADAR1, ADAR2, because there are long definitions of these enzymes are already present. That's why we are using the short forms only. Okay, RED2, ADAR3, or like this. These are the different sequence length of these enzymes. Okay, you can go for it. And this is one example of RNA editing of apolipoprotein B mRNA. Okay, this has been very good example. I have extracted it from this lecture videos. You can go for the lecture videos for more information. This is your DNA sequence, that is 5 days to 3 days and 3 days to 5 days. If you see here, this is a sequence CAAGTT. This is already present. Okay, in case of liver, unedited mRNA is present, that is CAA is present, that will lead to apolipoprotein enzyme with 4563 amino acid residues. Okay, understood? Next is transcription and splicing out of intron sequences. In case of intestine, earlier we have been discussing about the effect of RNA splicing in case of your calcitonin enzyme, right? Calcitonin hormone was there, which has crucial role in thyroid, but also calcitonin gene produces another enzyme or another hormone which has CZPR, crucial role in brain. So one gene producing two enzymes or two hormones which has two different functional roles. Got it? So here also this apolipoprotein B mRNA producing two proteins. One is for liver, another one is for intestine. Okay, for liver it has this apolipoprotein has 4563 amino acids. That means too long. Next is intestine RNA binding deaminase. Here RNA editing is happening. Okay, so RNA binding deaminases are already present. RNA editing oxidative deamination of cytidine. So we have been discussing that when there is a deamination process occurs in case of cytosine, that means your ammonia group or NH3 group have been removed. That will lead to production of your uridine or uracil. Okay, so this C have been replaced with uracil or uridine. Now it has become UAA, and everyone knows this edited mRNA UAA acts for a stop codon. Understood? That UAA, UAG and UGA, these three are stop codons when in the translation process. So after translation, it is producing apolipoprotein with 2153 amino acid long, which is approximately 50% of the previous apolipoprotein. So this apolipoprotein has more than 4000 amino acid, and it has only 2000 amino acids. Because of this insertion of one stop codon, we cannot call it insertion, we can call it as base modification or RNA editing. Fine. So this RNA editing been happening here using your deaminase enzyme. Okay, this is a very good example of your RNA editing using base modification process. Okay, earlier we talked about insertion and deletion of uracil. Okay, And now we discussed about base modification in case of RNA editing. And remember it, in case of base modification we don't require any guide RNA, but while we are inserting or deleting certain regions using this RNA editing process, we require a guide RNA, and this guide RNA has certain regions which is complementary to your previous mRNA or to the target site. We can call it as a vicinity to the target site, nearer to the target site. Fine. Okay, so today we discussed about alternative splicing, transhistorification, lariat formation, different types of introns, and uh, what is the function of these enzymes and all, and also talked about RNA editing method mechanism, including RNA editing enzymes, two different methods, that is base modification, and another one is your uh, insertion and deletion of uracil. Okay, so question 8, okay, I'm repeating it again. Priyadarshini, you can write a mail to the NPTEL team. They can answer this question for you. Okay, you can write it or you can just mail them that you have not submitted the assignments. What can be done? Okay, write them. Fine. Yes, here any base in an RNA can undergo editing. Yes, but this is actually it can be true or can be false. But I would suggest that it is false. Like any base cannot undergo editing. Okay, there are specific uh, processes already required for it. For example, your base modification and uracil insertion and deletion been happening. So postpone of exam. Okay, you can ask this question. You can write a mail to the PM sorry NPTEL team so that they can answer this for you. Okay, you can write a mail. You have been getting mail notifications, right, about different types of feedback or about different exams and all. So just write them. They will answer this question. Okay, regarding your submission of assignment or and about any exam or anything, you can write a mail to them. Fine. Okay, so now you have discussed everything about the week 5 lecture videos. You can go for the week 5 lecture video so that you will get a clear idea about what have been taught. So if you have any question, then you can ask me. Okay, then uh, let's end this class now. We'll be meeting on next Monday with week 6 NPTEL live session. Okay, thank you for joining.